All right, so on to the second part. How can we quantify the amount of DNA that we started with? And this is going to be useful for particularly for the field of expression studies. So when you want to know how much of a gene is being made, you can use this to quantify the amount that you started with. And we're going to quantify by doing PCR. So this is quantitative PCR. So let's go. All right, so this is set up as a little thought experiment. So you're supposed to imagine yourself in a PCR reaction tube. Every round of PCR, we are going to double the amount of DNA that we had. And so let's say we're at cycle 25, just arbitrarily. So, all right, what's in our tube at cycle 25? Yeah, a bunch of nucleotides, primers, template, amplicons, which is the DNA you just made, enzyme, magnesium, buffer, water. I think that's everything. Taq polymerase, I don't know if we didn't already say it. And so let's say we've got about 1 million copies of the amplicon at step 20 after 25 cycles. And so you could work that back, just keep dividing by 2, and you could figure out roughly how many copies that we started with. But that's not the exercise. Here, we're just going to say this is where you're starting. So let's just accept this. So if you got 1 million at cycle 25, how many did you have at cycle 24? Well, it's going to be about half the amount that you have now because it doubles every cycle. So you'll have about 500,000 at the previous cycle. And at 23, you'd have about half of that, 250,000. What about cycle 22? You'd have about half of that, half of 250,000, 125,000, just like that. Because, again, you're doubling every single round. That, I think, makes pretty intuitive sense. And now, what if we went past cycle 25? So if we have 1 million copies at cycle 25, how many are we going to have at cycle 26? Right? We're going to probably have 2 million. Yeah, just like that. So yeah, if you were to make a little graph of how many we have at 25, you see we have 1 million here, and at 24 we would have 500,000, so that's right about there, and 23, 22, 21, 20, just like that, And this is a graph that's going to look like a straight line if you show it on a log scale, and it's going to look like a curving line if you show it on a regular, just linear scale. So yeah. At cycle 25, you got 1 million copies, fine. What are you going to have at cycle 26? We just talked about this. You're going to have 2 million copies, right? Of course. Cycle 27, 4 million copies. You just keep doubling. And PCR, when it's running at its best, is just going to keep doubling every single round. But of course, doubling leads to a very rapid expansion in the number of products here. In this case, the number of amplicons. Remember, the amplicon is the DNA that you have copied. It's kind of the template that you've made into copies. Yeah. So cycle 26, 27. What about cycle 200? You would have so many amplicons. You would have, 10 to the 35th power tons of DNA, about the size of 10 billion Earth-sized planets. Yeah. So you can just set up a PCR to run for 200 cycles, and why doesn't that end up consuming the entire solar system? Great question. The answer is because you're going to run out of material, right? And that's fine. Yeah. So, it wouldn't fit in the PCR tube anymore if you had that much. And so, thankfully, exponential growth does not go on forever. We are going to eventually run out of material to feed into our reaction. But as long as we start out with a known amount of material, and we put the same amount of material into every single tube, and the same amount of Taq polymerase, the only difference, we're going to get to the same level. We'll eventually end up at maximum amount of PCR product, unless we don't have any template there, in which case we'll just stay at 0 forever. But we will get to the maximum amount at different times, depending on how much we started with. Will basically run out at a different time. It's just like if you had a bunch of people that can run at the same speed, and one starts close to the finish line, that person's going to cross first, and then the one that started next closest is going to cross next, just like that. And that's how this works. So in reality, if we were to plot out what happens after cycle 25, we're eventually going to top out something like this. And so there will be a point at which we have risen above the sort of background level and we can detect our DNA. And there will be a point at which we level off and essentially can't make any more DNA because we ran out of nucleotides or Taq polymerase or something. Yeah, we're just out. So how do we use this to measure DNA? Well, just like I said. So imagine you have now two different reactions. Imagine you have two people, one starts with 4 times as much DNA as the other person. So if one person starts out with 250,000 copies at cycle 23 and the other person has 1,000,000 copies, 4 times as much as cycle 23, then at cycle 24 you'll still have 4 times as much as I do, and at cycle 25 you'll still have 4 times as much. But if you look at when they pass a particular point, there will be a difference in cycle time. So you would have 1,000,000 copies way back here at cycle 23, but it would take me 2 more cycles, 2 more doublings to get to the point where I have 1,000,000 copies, because we double it every round. And so if you start out with 4 times as much, you're going to get to any particular point 2 cycles before I get there, because each cycle is a doubling. And 2 doublings, 2 times 2 is 4. If that makes sense. So if you were to graph this out by the number of copies, you can see that we're going to start at the a very low amount, but 4 times different. But you can't really see 4 copies of DNA. You can only start to really see DNA when you get to millions of copies. And we're going to end at a similar spot when we basically run out of raw material. And the big difference is going to be when we rise up. And so if you have more DNA, you are going to rise up and pass that arbitrary point, 1,000,000 copies, earlier than I will, because I'll have to wait 2 more cycles until I've doubled and doubled again until I get to 1,000,000 copies, just like you have. And so that tells us that the red tube, my tube, had 4 times less RNA than the green tube had. And the green tube's over here, if anybody is colorblind, or I'm talking about the left tube. Yeah. So, yeah, what if you started with 8 times less DNA template than I did? Fine, that's easy. Can you figure it out without looking at this thing? So 8 times less. So that means you would have to double once to get 4 times less, and then twice to get 2 times less, and then 3 times to get to the same amount as I have. So just like this, if I start out here at 1,000,000 copies while you still have 8 times less, then you got to double it once, twice, 3 times, so 2-fold, 4-fold, 8-fold until you're at the same level. And so a difference of 1 cycle is going to mean a 2-fold difference in the amount of DNA. Difference of 2 cycles is going to be a 4-fold difference. Difference of 3 cycles is going to be an 8-fold difference. Can you guess what a difference of 4 cycles is going to be? 2, 4, 8, 16. Yeah, it's going to be 16-fold difference in the amount of starting material there. That's it. That's the secret of quantitative PCR. And so, boom, just like this. If you have less DNA, it's going to take longer to get up to 1,000,000 copies. And so your curve is going to be shifted over this way. If you have more DNA, you're going to, you may start at 1,000,000 copies, in which case you just go up right there from cycle number 1. And you would crash out, probably at about cycle 3 or 4. And, that'd be great. But, yeah, it's fine. It would tell you had a whole lot of DNA to start with. All right. So, yeah, the quantity is going to be roughly proportional to 2 to the power of the cycle time, or cycle quantity. Sometimes they'll say CT for cycle time, or CQ is cycle quantity, number of cycles. Yeah, basically. So, yeah, here we started with 2 to the 23rd, or here we had, I had 2 to the 23rd. Here we had 2 to the 25th. Let's see, did we get that right? No, it's actually the inverse of that. So the quantity at any particular cycle, there we go, is 2 to the power of cycle time times the number that you started with. There we go. Sorry, I had that backwards at first. Anyway, you can plot these cycle time things on a graph, And you can relate those to the amount of quantity and the amount of starting quantity. And as long as the starting quantity is on a log scale, you get a nice little line that you can draw through your points. And that is what we call a good standard curve. So you're going to make a standard curve usually if you're trying to calculate how much of a thing you have. Because for any given PCR, you're using different primers. And it would seem like a given prime, a primer should always bind, but each primer is a little bit different. And so for any PCR, we have to run a standard curve, which is just doing the PCR on known amounts of DNA. And so you'll start with a known amount, and then you'll start with different known amounts, make a nice standard curve like this, and then you'll test out your unknown. And as long as your unknown is somewhere within the standard curve, then you can measure very exactly how much DNA you started with. There we go. So there's a good looking standard curve. Look at that. The little dots are pretty much right on the line. And the R squared tells you that that line goes pretty much through the center of the dots and doesn't really mess around much. So that's fantastic. Yeah, if you do it right, you could measure even a single copy. In practical terms, usually somewhere between 50 and 100 copies is around the minimum amount you can reliably amplify. 200 copies of, or 100 copies of a 200 base pair gene is a really tiny amount of DNA. Yeah, 2 times 10 to the minus 17 grams. Real small. So 2/100ths of a microliter of blood equivalent to. So yeah, very tiny amount. Okay, so how do we measure the amount of DNA in our reaction? Why? By putting in a fluorescent dye that doesn't fluoresce when it's just floating around in solution. But as soon as you've made double-stranded DNA, it's going to bind to the double-stranded DNA and it's going to fluoresce. And so you're going to put enough dye in there that could bind all the DNA that you can make inside that tube. So that it can bind the maximum amount of DNA that you could make in a reaction. And that's fine. These dyes are pretty cheap. They are what is called intercalating dyes, which means that they will sit in between the two strands of DNA. They will actually bind to the DNA double helix and kind of slot in there in the groove of the double helix. And when they are bound in just that way, they're now going to be connected up, hydrogen bonded to some of the atoms making up the DNA backbone, and they will then be able to fluoresce. And so these dyes are chosen because they have that property, that they are not fluorescent or very weakly fluorescent until they're bound to DNA, and then they become hugely fluorescent. And so you can measure the amount of DNA in your reaction by putting in this dye, and then after every cycle, you can shine a little laser. So this is our wavelength of light coming in, and you will get fluorescence back from all the dye that is bound to DNA. And so you can just watch the DNA accumulate. And so one of these popular dyes is a thing called cyber green. It's a thing called a cyanobromine dye. And yeah, that's actually spelled correctly down below. That's pretty good. Cyber green chemical is one of this family of dyes. These are commonly used. They give a nice bright green fluorescence, and they become much more fluorescent once they have bound to DNA. So you'll get a certain baseline level of fluorescence, but once they're stuck to DNA, boom, they become hugely green fluorescent. And so they'll tell you how much DNA you have inside the tube. The molecule looks like this, like so. The molecule sort of unfolds and slots into the DNA helix. It's actually going to bind in, I believe, the minor groove. You can see you've got the major groove here, where it's a farther distance between this strand and this strand. And you got the minor groove where they come a little bit closer together. And so our little dyes actually slot into the minor groove and bind right there. Now it says low toxicity up there, but it's not zero toxicity, because anything that can bind in the minor groove or major groove of DNA can cause mutations by binding to the minor or major groove of DNA. And so these mutations can build up and can potentially lead to cancer. But not all dyes are created equal. Some dyes, like this old one called ethidium bromide, shown here, causes a lot more mutations to happen once it binds to DNA than something like a cyanobromine dye. So they're much better, but they are not perfect. They're not zero mutagenesis, but they are low mutagenesis. All right, so CyberGreen in action. How's it work? Just like we said. You're going to have your templates. You're going to go through all your stages of PCR, your denaturation to separate the two strands, your annealing to bind the little primers down, and then your amplification, where the Taq polymerase is going to copy, starting with the primer, and extend those primers. And then as you make double-stranded DNA, as you extend those primers, the cyanobromine dye, the cybergold, is going to, or cybergreen rather, is going to bind in the minor groove there. And yeah, the more DNA you have and the longer the DNA is, the more cybergold you're going to bind. And the brighter your green fluorescence is going to be when you take a look at it in between each cycle. So there you go. That's how you quantify the amount of DNA. And so now the last step, the last part of our puzzle here is we've got to figure out how we quantify RNA. This is how we're actually going to look at gene expression, because you're going to have the same amount of DNA in every cell, but depending on how much messenger RNA you make, you're going to make a different amount of protein. And we want to look for differences in gene expression, or we want to look for the amount of an RNA-containing virus, like SARS coronavirus 2, that a person has, or like hepatitis C virus, or like HIV is an RNA-containing virus. And so for this, we have to do reverse transcriptase PCR. And that is also going to be super simple. Step one, we're going to turn our RNA into DNA by copying it with a special enzyme called reverse transcriptase. Step two, we're going to do PCR, just like we did before. Step three, we're going to read out the amount of PCR that we've done. And in this case, we're going to use a special technique. Instead of just having fluorescence that binds and sticks to our DNA at random, we're going to add in a special element called a probe. And so I'll show you that in a minute. And yeah, it's pretty neat. And this is how the highest end, best quantitative reverse transcriptase PCR is done. But that's in the next slide set. So let's go there now.